I quickly realized that AI, machine learning space, had the potential to replace everything I had learned at school regarding industrial engineering. So when I understood that, I made a deep dive. I went into AI fully. We were the first ones to be capable of predicting atrial fibrillation several minutes in advance based on onboarded wearable. Accidentally, someday, we wanted to test some new types of neural networks. We found a very interesting use case in drug discovery. It was like small molecule protein interactions. We took some data to build a model and test these new graph neural networks at the time. And we realized that we had something that worked super well. There's a couple of indications that are prominent throughout the population, but the 5,000 plus human diseases are mostly rare, meaning that they have very small markets attached to them. This means that there's a very small or low incentive for companies to put 2 billion of investment for a 2% success rate of finding something that can be used in only a couple hundred or thousands of people in the end. Many call it the one drug, one target paradigm. I call it a dogma because it's really almost a belief at this stage. Some people are convinced this is a reality, that drugs are designed to be extremely selective, potent on one target, and that this is what leads to in vivo effects. But there's more and more evidence that it's not the case, and that even making the drugs super potent on a single target and very selective does not correlate with more drugs showing activity in vivo settings. There was a very interesting study in which a group of researchers took the 300 most common oncology drugs, which had an identified putative target of interest, and then they genetically engineered the corresponding cancer cell line to silence the putative targets. So they removed the supposed targets of these drugs in the cell lines, and they tested the drugs again. And most of the drugs maintained their effect. So they remained cancer killers. Sometimes they've lost a bit of effect. They were slightly less potent, but they still were cancer killers, which shows that these drugs, supposed to be potent inhibitors of protein A, actually aren't, and they likely have a much more complex polypharmacological mode of action. We don't have to do in vitro large-scale assays anymore. We can just do the predictions and then do the minimal in vitro validations to double check that those predictions we've done are true. So if you have AI to explore drug-protein interactions and AI to explore all the pathways that we have already identified, it's much easier nowadays to say, okay, in the context of this cancer, modulating those three pathways at the same time has more potential than touching a single target somewhere. The data to solve drug discovery does not exist yet. If you want a model that has the capacity to generalize on a problem, you need highly heterogeneous training data. Kantify it's a tech bio with a mission to use AI to improve human health. What I believe differentiates us from the rest is the fact that we have created the so-called foundation model for a small molecule protein interaction. And we use it to do target deconvolution, so to explain which therapeutic targets can be modulated for various disease types. We also have a very large focus on rare diseases. There are two successful cases I can talk about. Sarcoma and LGMD2, so limb-girdle muscular dystrophy of type 2, where it was exactly that. We had some source phenotypic inputs, which were screenings of molecules with the notion of readout. These have some effects that is positive and all the others don't. We made our predictions with Sapien hits over the entire proteome. We deconvoluted a number of targets we believed were playing a role in the response. The partners validated those on the in vitro model through some silencings first, and then silencing plus retesting the drugs that worked. And suddenly the drugs didn't work, which validates the role of the targets. Every time we focus on a single target, it's harder to get an in vivo response. That's why we are starting to believe in polypharmacology more and more, because we see way more positive results in the polypharmacological approaches than the classical single target projects. The point where I beg to differ is on the impossibility to screen virtually for novel targets. That is with classical approaches where models do not generalize. The coverage in terms of protein and molecular diversity is enough so that now when we present like a new protein for inference, the model still manages to recognize some features in it and to propose small molecules that are going to bind to it, modulate it, whether activation or inhibition. Health is really the sector that has the most data online. If you want to solve problems, go in healthcare. There's like entire databases of millions of data points reported by tens of thousands of researchers that you can leverage. And we saw that some of it seemed to be underused or underperceived, and we just made use of it.